Probably me.
All right. Welcome everyone to our CLS talks. This is our March 2026 session. We're going to talk about CLS research. I want to introduce Jen Lee. Dr. Lee is the founder of the Consortium for Understanding Research and Education in X-linked Neurodevelopmental Disorders. That organization is Cure NDD for short. It's an international nonprofit that unites researchers, clinicians, patient advocates, and families to accelerate discovery, share knowledge, and drive innovative therapies for X-linked NDDs. Um, Dr. Lee has held several leadership positions across the innovative and venture divisions of pharmaceutical companies, health insurers, and major hospital systems. She earned her BA from the University of Pennsylvania and her PhD in experimental psychology from the University of Oxford. Jen Lee is a mom to a child, a daughter with Coffin-Lowry syndrome, and her family is committed to accelerating therapeutic development for this disorder. Real excited to have Jen present today and talk to us about some about advancing CLS research. So Jen, I'll pass the mic to you.
Yeah, thank you so much, Teresa, and thanks for having me and Mary present. So I have a few decks here for everyone, and thanks for taking your busy times to listen to me talk today. I was fortunate enough to meet two of you prior to this call. Um, but before I start, I would love for Mary to do a brief intro. Mary?
Sure. Thanks, Jen. So I'm Mary. I'm a geneticist. I'm from Alabama. I got my bachelor's, master's, and PhD from UA Huntsville. So for those of you in the area, and then since then I've been in medical affairs for a number of pharma organizations and started my own consulting firm a couple of years ago. And my focus is on neurodevelopmental disorders, particularly from a genetic basis. What is the genetic etiology? And particularly excited to meet you all after meeting Jen, and really excited to move this work forward. So thanks for having us.
Thank you.
Yeah, so Mary and I prepare a few slides. This is meant to be a discussion, so welcome to stop me at any time. I am, here's the agenda. So we'll do a brief intro of my background, and then I'll talk to you about what's important in terms of research as a CLS mom scientist. Um, um, so again, welcome to stop me, ask questions at any time. So as Teresa mentioned, I started telling everybody after my daughter was diagnosed, I was like, there's some reason in the universe that when I was doing my PhD, I was drawn to brain development. So I got my PhD at University of Oxford, England, where my PhD was actually focused on putting electrodes on babies' heads. So these are, I've tested over 100 babies under age 18 months, from birth to 18 months, to correlate their uh, uh, brain stimuli, evoke potentials, that's what we call it, brain waves, to their what's considered a normal brain development. And then after my PhD, I've worked in hospital systems, house insurance, and pharma. But I think the most important thing I've done was a company that convinced general pediatricians to do developmental screening. So this was back almost 10 years ago now. The Lancet actually wrote us up, and I was fortunate enough to do a TED Talk. So you can take a camera out, and the QR code will go to my TED Talk. Basically, just outlining how important it is to get a diagnosis early on, so that we have a fighting chance for our kiddos. But I tell people, especially for this community, it didn't matter what my credentials are. What matters most is I'm the mom to this beautiful soul. So about half a year ago, she was diagnosed with Coffin-Lowry syndrome. Despite all my connections, it took me 6 years to get diagnosed. And part of the reason is because she's pretty high functioning. She don't have, the seizures and the drop attacks. But I knew something was off. And yeah, I had a very hard time convincing people until finally we had a exome sequencing done on her. Since then, I've done a few things. So one of which, as a mom, I need to let her understand, why she's different. So we publish a book called Metro X. Welcome to use a QR code again, and it's a children's book, easy read, basically saying, despite what she has, she's unique in her own ways, and her X is a play on words for the X-linked in the genome that makes her special. And then, as Teresa mentioned, I did also create a international nonprofit called Cura NDD. These are my girls designing the poster. It's meant to be a consortium for multiple X-linked neurodevelopmental disorders to come together, drawing families, researchers to really understand, what's going on biologically, as well as advancing research for our kiddos. And I think that's why I've been telling people more than full time since we're only about 4 months old. And yet we have 11 different foundations now as part of our consortium, and the number is only going to grow because on the X chromosome, there are multiple genes that we know have diseases associated with it. So unfortunately, this number will become bigger as there's more whole genome sequencing done and so forth. And it is international consortium, and Coffin-Lowry Foundation is part of that consortium. Right now, we have members in UK, and I was talking folks in Middle East and so forth. And like I say, welcome to stop me at any point. Otherwise, I'll just keep going. When my daughter was diagnosed, just having done what I done, there were so many questions I want to answer, and I couldn't because there's so little research, as many of you know. So the first thing I did was reach out to CLS Foundation, and Teresa was very kind to walk me through what's the support, and I was able to connect with Annie. I'm also physically based in the Bay Area, so right now it's like 90 degrees here. Um, um, so thank you so much for everything the foundation have done to date. But in terms of research, there was a lot of gaps. The first question I asked myself was, where are these patients are? And when I asked this question to Teresa, she sent me this report, which I believe many of you have seen. She presented this in June 2025, right, Teresa, in your last family meeting, where there's about 300 families in the US and then about 500 families worldwide. And I believe that number probably have gotten bigger now.
Yeah, it's increased. This last newsletter we mailed 540 out. So there's probably about 50 more. And it's pretty much a 50-50 split between international and US, roughly.
And me being a scientist, I was like, that's amazing. We need to demonstrate that there is a need for CLS by demonstrating the numbers is big in order to get support from researchers, in order to get interest from investors, in order to get funding coming in to help our kids. So what I've done is I reach out. Mary actually been in this space for a long time, and she and I both, not knowingly, reached out to the same lab test, and who was kind enough to present their data. So this is data from a single lab test company, and this is by far the largest lab that has most of our kiddos, because in their count as of October last year, they have about 200 kids, right? So which is about 2/3 of our kids that Teresa have been counting. And as you can tell from their report, there's a little bit more female than male. There's a lot more European who's affected, But what's interesting is most of these kids who are getting diagnosed are now around age 2 to 5. Unlike my daughter, who was diagnosed much later. But there are a significant amount where there's also being diagnosed, past 18. So that means, especially for girls, as many of you right, they are asymptomatic until they have kids and so forth. And the other thing that jumped out on me on this report is in 2021, they only have a count about 100, and within 5 years, that number doubled. So we have no doubt as genomic testing becomes more widely used and there are more people aware of CLS, we're going to get our numbers up because there are more kids out there
Yeah.
that we need to find them. Anything you want to add, Mary?
Yeah, Jen, I've got a question.
Yeah,
What was this Gene DX, this
that's
lab?
right.
Okay, all right.
I was going to ask the same question because I work for GeneDx, ironically.
Oh,
So
what do you do, Jen?
I work in the clinical genomics department.
We have a lot to talk about.
Okay, great.
Yes.
You might be able to see all my zebras in the back. I've been there for about 14 years.
Oh my
Oh
goodness.
my God. Yes.
But I'm sort of I'm like a test manager, program coordinator I sort of make sure things don't get stuck and tests aren't late, to put it
No, you're my new best friend. We have a lot to talk about.
Okay. Keep
And you'll
going.
see why. You'll see why in a minute. But Mary, anything to add on this?
Not on this. More to come on GeneDx data.
Okay. So GeneDx is a longtime partner now of Mary and now me in all my 6 months of doing CLS work. So remember, I've worked in hospital systems and health insurance and pharma. The next question I will want to know is where are these doctors treating our kids? I need to know who's our geneticist, who's our neurologist, so that we can ask them for expertise. We can see if are they seeing more than one CLS kid, and should we be calling these CLS experts out there that we don't know of, right? So we can only do that if you, meaning listeners here and out later here, please email us. Please email Teresa who's your doctors, because we want to identify later on maybe center of excellence, but we need to know who's doing the work, who's treating our kids, so that we can get more researchers on board. And you'll see this consistent thing because we cannot do this work alone. We need all of us to come together in order to provide the data on our kids, on our doctors, generate more interest so that we can advance more research and maybe find a cure within my lifetime for our kids.
Hey, Jen, can you go back to the chart that shows the numbers like per state? One more. Okay.
Yeah.
So, the problem here is, so let's just take Alabama, for example. We have 6 people in Alabama. Now, there are probably 4 of us in North Alabama. So we could go, hey, any of us seeing the same geneticist? But just because a geneticist has, let's say, 2, maybe 3 patients with CLS, it doesn't make them an expert in CLS, right?
It sort of does though, because the numbers are so rare, because it's such a rare disorder. Even if a physician just has 2 or 3 patients, they have 2 or 3 patients, and that does make them something of an expert when in such a rare disease. And if we can take somebody like that has some experience and then help them to gather more data, they can end up championing this disease from the physician side. So, a physician with 2 or 3 patients is completely worthwhile to share, for us to reach out to ask them, how did you diagnose this? Because they've had a few, and that's enough for it to be meaningful.
Okay, all right. But so the geneticists that I know of, there's just a few of us at this like 30-year age, and then there's some babies. Completely different geneticists did the diagnosis, right? Because they're the one we saw is still not around. So they saw other people, but I don't know, my neurologist or Matthew's neurologist, I think Matthew's the only patient with CLS that he sees.
Yeah, it's just so unfortunate, like because it's an ultra rare condition, right? Like once a physician sees more than two, they become an expert. So the next thing we want to do is can we interact with these physicians and foster more interest? So Mary and I, you'll see in a bit, like we're actively going to conferences, going to academic centers, going to academic meetings, right? Trying to advocate and teach them about CLS. So
Yeah.
we need them and those people who have seen it will know what we're talking about. Um, the next thing that Mary and I are working on is partnering with general pediatric clinics. So we're doing a national 3 month pilot with Gen Peds to teach them and help them implement to prescribe more genomic testing, and that could be exome or whole genome. Because one of the main reasons that my daughter took 6 years to get delayed in diagnosis was because the GMPs had no idea, nor did they even think of doing a genetic testing, even though American Academy of Pediatrics last June recommend pediatricians to do genetic testing once they suspect intellectual disability or autism. Um, yeah. Anything to add, Mary?
Yeah. So, as you all know, you probably all went to your pediatrician first, with delays and issues. And as Jen said, despite the guidance from American Academy of Pediatrics, these pediatricians just don't know how to do it. And it's not that they, are unwilling. They weren't taught about genetics in medical school. They're already very busy. And so basically what we're going to do is just sit with them and along with a number of other sponsor partners, to provide them a lot of resources and help them understand the importance of genomic testing, the diagnostic yield or clinical benefit of genomic testing, and then just practically show them how to do it. And it won't take long for them to get it, and then start implementing it. And then we're going to present those findings at a national meeting for pediatricians, and then hopefully more pediatricians will start adapting this and getting more kids diagnosed, not just with Coffin-Lowry, but with a number of developmental disorders much earlier. So we're very excited about this. Jen is doing a wonderful job of talking about this program and soliciting sponsors for the program. So we've already gotten a $50,000 commitment for funding from one company and at least a $20,000 commitment from another company. So we're well on our way to getting this off the ground. And we've got a number of pediatricians' offices right now filling out surveys on what their current progress is, what their current protocols are, and if they're interested in participating in our pilot. So it really could change the face of medicine as we empower the general pediatrician to start testing for developmental delay.
So my ask here is, if you still have a pediatrician, please let them know about this and see if they're interested in joining the pilot.
Hey, Jen, we should
Yes.
probably get this call out in the next newsletter.
Yeah. I
I
think that's
think
good.
we reach the majority of the people there. I might could do an email asking for that as well, but we should probably follow up in the next newsletter.
Yeah, because this is going to be huge. This has never been done at a national scale. And we're looking at very different pediatric clinic sites. Some pediatric clinics are private, others are linked to academic medical center, and we're doing it purposely on various sites to help them understand, when they see a child with intellectual disability, their first go-to should be genomic testing.
Hey, Mary, would you be the person to provide me with the information to disseminate?
Yes, ma'am.
Okay, great. Thank you.
I forgot in the South, you guys say ma'am. You ladies rather. So my second question is, what's considered typical for patients with CLS. You can call that typical, atypical, what's the norm, but I want to know what's the future for my daughter. And we know all our kids, I've heard this from other rare diseases, they're like, our kids are like snowflakes. Every single one of them are different and fragile, right? That is true, but I do believe that we can find consistent patterns. But again, I'm a scientist, so I'm hopeful. So one thing we've done, thanks to Teresa, was you probably, you might remember all of the reminders about core registry and Sanford research. So because of it, I believe, I think there was 95 data inputs, was it,
Yeah, I
Mary?
think we're up to 100 now.
Okay. And then Mary actually dig into this data. So Mary, go ahead.
Sure. Yeah, so I submitted a request to receive the de-identified data from the CORDS registry and received all that data, and then there, deduped it. So basically there were a lot of duplications of the same patient. So I removed the redundancies and then really dug in deep into that data, and tried to make some correlations. And so importantly, I took this data in combination with some data that GeneDx gave us. So GeneDx gave us all the data on the numbers of pathogenic variants they had received in a certain time frame, along with the total overall number of tests that they ran for autism developmental delay. And then we also combined that with your data, on numbers of patients and where. And so basically, we took all of this data and integrated these data sets to assess for gaps and opportunities for drug development. And our goal here is to establish a scalable registry framework and define the priority elements to enable comprehensive characterization of the natural history and phenotypic variability. And so really the first part of this is taking all the data, here's where we got the data from, and putting it together. And then the next part is taking what we have and trying to identify, what's correlative. So, what do we see in a majority or most of patients that we can then try to test in future patients and see not only what's correlative, what can we say is normal for Coffin-Lowry patients, and then potentially what could we influence? So if we were going to start a clinical trial, what would be the natural endpoints in that study? What would be something that we would expect Coffin-Lowry patients to have an issue with, that we could also try to improve with clinical research? And this will be important as we try to get drug companies to start developing therapeutics for this. They'll need to know what are the endpoints for clinical studies that they can then measure their drugs on. So it's very exciting to start to see some of those correlations. So you see in the figures here, in the front middle, figure 1. So this is patient reported outcomes, where they are, the types of clinicians, mostly geneticists, followed by neurologists and pediatricians, and then what types of mutations. The bulk of mutations were de novo, meaning they were brand new. They were not inherited from mom or dad. And then, a minority in this data set were inherited mutations. Then we have the numbers of likely pathogenic variants, so variants that we know are causative of disease. We have the numbers on those. So what I've done before with similar data is I can take this data now with the existing published data and work towards a more accurate incidence prevalence numbers. And this will be important to know, again, for potential future drug development, how many patients there really are. So then on the figures on the right, figures 4 and 5 are just the start of analysis of our data set on some of the delays in these patients compared to developmental milestone times of other children. So you can see some of the independence things, such as using the bathroom unassisted, eating unassisted, holding pens, how Coffin-Lowry patients are behind developmentally to their peers. And then, self-help tasks, dressing themselves, showering independently. We can see the differences here. So we've done this work, and we've also done a lot of foundational work on what will be required for a natural history study, including additional collaborations with other organizations. So GeneDx has agreed to give us more and more detailed data to move towards an epidemiology assessment. And then we're working with other organizations. We've applied for data from other organizations as well. And then we're also doing a deeper dive into this to look at differences in sex. So, as you know, the boys and girls are quite different in their presentation and in their prognosis. And what's somewhat surprising is that the bulk of published literature just averages all the patients together, even though the boys and girls are quite different. So when the data are averaged, it results in neither one being accurate. The girls appear in the published literature more severe, and the boys less so. So we're parsing out this data, separating out all the boys and girls, and then reanalyzing all of them. So that's a publication that we're working on now. And then we're looking for additional genetic data and working towards a true natural history study that will combine genomic data with the phenotypic data. So the data that we have now is really just phenotypic data unmatched with genetic data, and we really need that. So that's something that we're going to work towards moving forward. Jen and I have collaborated on an application for a large NIH grant, and we'll be applying for more grants. So we're really coming at this from every angle. So we appreciate your contribution to the research, and we're really excited that we're finding things and hopefully getting pharma companies excited about Coffin-Lowry and excited about the potential for therapeutic intervention.
Hey, Mary, can you do me a favor and describe genotypic data versus phenotypic data?
Yes, thank you. And I apologize for not jumping into that right away. So basically, your genotypic data is your specific DNA. So when you send your child's blood in for genomic tests, they're basically looking at your A's, T's, G's, and C's and determining what is your specific change in your DNA that results in the phenotype. So the phenotype is your symptoms. So, the stimulus-induced drop attacks, or, sleep issues, or dental issues, or curvature of the spine, that would be your phenotypic data. And then your genotypic data would be, do you have a large deletion? Do you have a small deletion? Do you have a missense variant? The specific DNA variant that is causative of disease. And it's important that we match those, because different types of variants can commonly lead to similar phenotypic presentations. So, and that correlation just hasn't been done yet.
Yeah, let me ask a question though. If we're getting this, so the genotypic data is really provided when when our children were diagnosed, if they went through genetic testing.
That's right.
But some of these kids with CLS now are babies that are being diagnosed, because genetic testing is more readily available than when some of our kids were small. So some of those phenotypes have not yet presented.
Right. Yes.
There would be a mismatch there, right?
Well, basically, that data would be somewhat limited in utility for the moment. That being said, even if we don't have a phenotype, the genotype is still relevant, because we can still count that variant type. Sometimes once you have a certain threshold of data, you'll start to see common variants. So we'll start to see the same variants over and over. And ideally, over time, what we'll be able to do is take the matched genotype phenotype data, and we'll be able to provide predictive value for patients that have not yet fully presented their phenotype. Because once we have a certain threshold of data, we'll be able to explain better to those parents, based on this phenotype, your baby is likely to struggle with A, B, and C. Because we will have gathered enough data to be able to do that. And, it's knowledge is power. So
yeah, so I would think in that case, you would need the data from the kids, like my son and some of the other mothers
yes,
on here, that
yes,
age, to know what phenotypes did present.
that's
Um,
right. Yep.
but the sticky point here might be that some of the kids, or many of the individuals in their 30s and 40s who have CLS, didn't have genetic testing, so they don't have the genomic data to use in that variant identification.
How were they diagnosed without genetic data?
Characteristics. We just recently got Matthew tested, and his variant identified. I didn't have it before a couple of years ago.
Well, I mean, now you do, though. I mean, it's never too late to get that
I do. However, many of these families do not have that data, I
right.
guess is what I'm getting at. Judy?
Yeah, my son is 42, and at the time when he was a year, going back to about 1985, there was no genetic testing. We went Ian was seen at Stanford. Stanford looked at his hands and our hands, looked at his facial phenotype, and we were brought into a hot airless room at some point and given the diagnosis of possible Coffin-Lowry syndrome, which meant nothing to my husband and myself. But that stayed, and then at one point, Stanford took photos of our hands, Ian's hands and all of us, and there was no film in the camera. So Ian was never officially had the diagnosis of Coffin-Lowry syndrome, but he definitely has Coffin-Lowry syndrome. But it's all based on non-genome, I guess is your phrase, but just phenotype and shape of his hands, those characteristics.
Your data, Judy, are still super valuable because you've still got history, you've still got, you still have important phenotypic data. What you have is a clinical diagnosis, and not every neurodevelopmental disorder has that capability, but because you do have it, I mean, it's still super valuable data. We still would love to have your contribution on this is how we progressed, this is what age we saw the first symptoms. It's still super relevant. Christy?
So my guys were a little different too. So actually my guy's blood work went to France because that was the only person doing any research at that time. So the only information we got other than facial and hands and all that was they came back and they said that the boys were located on the bottom part of the gene, but they never gave us anything officially other than a verbal thing. It was a different part of the gene. They didn't understand why, but we were never given any specific paperwork to that. That's just what we were verbally told that backed up the initial diagnosis of the facial features and stuff, because the doctor that had actually seen Anthony and Timmy back in the day had actually saw one other CLS child. Which was interesting. So we have, just like them, we're part of the older population and we have parts there, but there's a lot of parts not there in the sense of the genetic testing.
Yeah. Hey, my son was also in that same age range as Christy's boys, and we also sent blood work to France. Dr. Hanauer is the doctor who actually, I think, identified the gene, was doing studies on it, and we got an email from him that says Matthew has been identified as having the defective gene associated with Coffin-Lowry syndrome. And that's all we got. That was good enough for me, though.
Yeah.
That was more than I got. We just got the verbal from the doctor saying that, or from Greenwood Genetic Center saying that that's what the boys had.
I think this is good info. I think one thing for us to know, one thing Mary and I can work on toward is maybe getting some funding to get the older patients diagnosed, get that genetic testing done.
Yeah. If y'all would be interested in that.
Yeah, we recently did it. So I know, Jen, you've got Matthew's report. And it was not a blood test, by the way. It was just a saliva. They did a cheek swab. So it was a very easy, for us anyway, the lab that we used.
Was that something you guys did on your own, or was that something a doctor recommended, Teresa?
We went to a genetic clinic here.
Did y'all
Yeah,
have
typically,
or Huntsville Hospital?
No, it was through Hudson Alpha Smith
Oh, great.
Family Clinic.
Yeah, that's awesome. They're really good.
Mhm.
so Christy, given as mentioned, Mary and I is working on promoting this genomic testing prescription among Gen Peds. I think general pediatricians, if they know how, should be able to prescribe you that testing. If not, geneticists can definitely do so. But usually, medical geneticist has about anywhere between 3 to 6 months wait. So that's why Mary and I has this initiative to get more Gen Peds more equipped to prescribe the whole genome testing. But
And then the other side of it is possibly a lot of our older population. I'm sure insurance is not going to get paid to get retested for something they already know that they have.
no, actually, on the contrary, I used to work at health insurance. If there's a clinical suspicion, which it sounds like you do, of CLS, it gives them the reason why they should get that genetic testing.
I guess it depends on how. Yeah, because I don't know how they did that all the first time around. Other, like I said, they just worked with. Yeah.
And we don't necessarily need everybody's samples. We just want to make sure that y'all know that this is the work that we're doing, and we want to provide updates on the research that we do as it comes out, as it's super fresh. This poster that Jen showed, we just presented it last week at the American College of Medical Genetics. It will be published in Genetics in Medicine, the journal of the American College of Medical Genetics. And then the Meditech Today, just an online journal, published a paper about our collaboration with CureNDD and Biologic and Northwestern University on the work that we're doing to advance research for Coffin-Lowry. So, the more we can publish about this, the more we can raise awareness about the disease, the better chance we have of more kids getting diagnosed and pharma and biotech companies starting to get interest in potentially pursuing therapeutic interventions. So, every bit of awareness and raising this progress and these questions publicly will help research efforts.
Yeah, and I do want to, thank you for everyone who contributed to CORDS, and especially to everyone who took that extra 30 minutes to speak with me, because it's just do take a village. Like, I only have my child who's not typical, because she is quite high functioning, but she does have CLS, and we want to gather those stories and gather those data points so that we can present to what is considered the norm. Okay, I have a few more slides.
Hey, Jen,
Yes.
one more thing on that while we're on the CORDS database. If anybody on the call has not submitted a record for their family member, please do so. And if you have, they're asking for updates every year. So if you've had any change or you get a report or whatever, please go in there and update that.
Hey, Teresa, this is Margaret. Just a quick question on the registry, or maybe this is for Mary. Can y'all hear me?
Yep.
Yes.
Yeah. Okay. Just wondered, I mean, I'm sure you're familiar with Florida doing the genetic testing of their newborns, and I think it's a pretty big project that they're undergoing. Is there a way to connect with, and I don't know if other states do that also, but is there, and I don't even know if they test for CLS. I think they test for a lot of, rare genetic diseases, but is there a way to connect with sort of political, states at that level to get information from them, or to even at least to provide information to parents if their children are diagnosed?
It's a lovely idea. Jen?
I'm 6 months in. I think I think it's a great idea. So to my knowledge, in order to get on the newborn panel test, there has to be a therapeutic.
That's right.
Without a therapeutic to CLS, we would not be on that newborn panel.
Yep.
Like regardless how much lobbying we do, which is why it's that much more important for the community to come together, right? Pull our data, pull our awareness, tell everyone about this so that there's more interest from investors, from pharma, from people who has bigger dollars than us, to keep this going.
Yeah. I also had a discussion with Dr. Wendy Chung at Boston Children's this past week at American College of Medical Genetics. She's running a similar study in newborns where they're doing basically whole genomic testing for all newborns. So they've got 25,000 babies that they've already sequenced, and they're identifying different things far earlier than normally kids would get diagnosed. And then they're able to intervene, prior to kids showing symptoms, which is really neat. But I have spoken with her about searching her database for any Coffin-Lowry patients, and so personally, I'm not familiar with which data are being gathered in the Florida initiative, but if it is a genomic newborn screen, chances are what's happening is they're sequencing every gene, but they're only analyzing a subset. So what we can certainly put that on our list of folks to circle up with and see if they are in fact sequencing, our specific gene, and then see what it would take for them to share that data.
Yeah, the other thing I do, Margaret, thanks for the question. Is I forgot to put in the slide actually, is I'm working with initiative that will put ICD-10 codes to be associated with CLS. So ICD-10 codes are what clinicians use to reimburse. So when the doctors see you, they need to label this code in order to get reimbursed from health insurance. Without that code, we won't know, right, how many physicians out there who have seen CLS. They could label it as intellectual disability or something else, or seizures, right? So in order to get that effort done, it has to be gone through CDC. CDC is unfortunately not taking any more codes right now, but there's initiative with other consortiums that we're part of that is trying to pull together a whole bunch NDD neurodevelopmental disorder codes to go in a batch. So we're working on that because that will also be huge to help us find where are these patients are.
Mm-hmm.
Yeah. Cool. So two more questions that I'm looking to answer as a mom scientist is what are the treatment options for CLS? I think a lot of the parents here who have older CLS kids, you probably know this better than I do. The treatment options are very limited, and it's a cocktail of drugs to address the symptoms, not the cause. But having worked in pharma, Mary and I, we also know that drug discovery is very long. Typically, it takes 10 to 15 years, $1 to $2 billion. Okay, so it's not an easy route, but I could tell you we're very, very much committed to find a cure within my lifetime for my daughter and her kids. And this is what a typical drug discovery look like. We start with molecular assays, then we try to map what CLS will behave like in a petri dish. Then we develop animal models. So usually, once we have compounds that drug companies think it might work. They will test it in mice and then see what happens in mice. And sometimes FDA or mandate to also go into non-human primates, which means usually monkeys. And then finally, they will submit into a process called IND, and then that's when it gets to clinical trials. Typically in US, there's 3 phases of trial, but for rare disease such as ours, we're looking at phase 1, 2 combined, and then phase 3. So phase 1, 2 is about safety, and then phase 3 it's about efficacy. So that's sort of the process we're looking at. Anything to add, Mary?
No, not on this one.
Yeah, so I just want to highlight, it is a tall mountain, but we are determined to climb that mountain. And we can only climb that mountain with all the help of you guys. Because all this requires, endpoints, the data to figure out what symptoms we can go after, what's considered significant change for our kids. Um, and then in order to do a lot of the cellular work, we need to model what CLS will behave like in a petri dish, in a mice. And that requires what's called biorepository. So typically, I think Teresa have mentioned this a couple of the family gatherings, it means a blood draw. Sometimes it could be a skin punch, so skin graft. And then the scientists can reprogram some of those cells from your blood into whatever cell that's here. So in here, it could be a neuron, it could be a cell in your liver, it could be a cell in your heart, whatever the scientists need to find the questions for. But it requires blood draw from our kiddos in order to model those disease state cells. Um, so there are biosample collections happening that Cure NDD as a consortium have engaged with. There's one happening actually in Mineral Park for Judy and Anne. That's on May 1st. There's also several others that are happening around the nation. That's another aspect that we're looking, to get funding for so that it will be free for your family to come and donate. Because as you can imagine, storing and reprogramming these cells are not cheap. It requires constant refrigeration and biobanking, but it's absolutely necessary to get done in order to figure out, what drug, what compound is effective in our kids. And then something Teresa and I have been talking about is if I play my cards right, hopefully by the time that we have our 2027 family gathering, I could bring some scientists in to do some of the biosample collection at that point. But more to come later.
Hey, Jen and Mary, let me try to kind of simplify the way I'm thinking about this is so we've been researching some bio sample repositories which would store this bio sample, whatever it is. Blood, I don't think can be stored as long as maybe a tissue sample, like that punch biopsy you mentioned, Jen. So the way I was looking at it, and there's some already out there, there's some repositories already out there, and there's some repositories out there that have CLS samples. Hudson Alpha did a research last summer, and I did include a copy of the paper that the researcher did on the website and in our newsletter on CLS, and they actually got that sample from I think it was North Carolina where it came from. But anyway, so it's already out there. You can Google it and find out what they have. But what we would do was we would help facilitate the collection of the samples and then put them. Then they would be housed and stored in this repository for researchers to access that repository. Researchers from all over the world could do that, right? They could go to the repository and say, hey, I need as many CLS samples as you have, and then that researcher would likely purchase those samples, and it wouldn't be the entire sample, it would be a portion of it, and
That's
then
right.
the sample still stays there for the next researcher. Did
Yep.
I kind
Yes.
of
You nailed it, Teresa. That's exactly how it works.
Okay, that's the way it works in my mind, but I wasn't completely sure.
No,
So we are
thank you for translating.
okay. So we are researching some of those repositories right now to find the most cost effective, because again, we're not a organization that has really deep pockets. So one that is the most cost effective and the one that would be the most bang for our buck long term, because we don't think this is going to be a quick discovery or an easy fix.
Right.
Yet, I mean, we need to plan to be in this for the long haul.
That's right.
Okay.
Yeah. So we're having several conversations, but that is just part of the drug discovery process, and it's not even for drug discovery. It's also necessary important for biomarker studies and endpoints. Basically, it's a way for researchers to figure out, is this vitamin making a difference for CLS. In order to do that, we need to see it at the cellular and molecular level, which is why those blood donations are important.
And is it also true that the biomarkers are key in order, I mean, those have to be determined before any pharmaceutical drug company would even consider a
Not always.
okay.
Biomarkers are super helpful when you have them, but even in the absence of a biomarker, you can still develop a drug. It just having a biomarker can accelerate the drug development.
Okay, all right. No, that helps. Thanks.
Yeah, thanks for that. So my last question is, as you can tell, this is going to be a long row and it's going to be an expensive row. But my family is absolutely committed to do this because this is a club. I did not ask to join, but hey, I'm here, right? I'm sure all of us love this moment. Having come from, done this work decades ago and working in healthcare, my biggest contribution is how can we get other people to become excited, to do research, to pull amazing people like Mary to come in and work on CLS. So Mary are busy. We're going around the nation, basically telling people, hey, we're doing work in CLS. What CLS is, it's a tremendous unmet need. Here are the data. We have natural history happening, and there are more stuff coming, such as sample collections and so forth, trying to get people excited. And the reason I put in here all my dates is, if you happen to be in any of these sites, I'm more than happy to meet you in person, shake your hand, talk to you about my daughter's condition, your daughter, your kids, and answer any of your questions. But there are more events coming up. So if you take your camera and go into this QR code, we regularly are out there educating researchers, clinicians, academics, get them excited to research on this. And this is also, as mentioned, it's going to be expensive. We need funding to create those mice models. We need funding to get researchers excited, to fund them to do the analysis on our data, on our biomarkers, to get the endpoints so that drug companies will be interested in developing drugs to help our kids and our grandkids. Drug development, as mentioned, on average, it takes 10 to 15 years, $1 to $2 billion. Only about 15% of drugs ever discovered in preclinical phase get to FDA approval. So it's gonna be a hard road but I believe it's not impossible. Because Mary have worked in several companies who specifically work in neurodevelopmental spaces, and I know we can gather a good team of scientists behind us to work on CLS. The other thing we're looking into is drug repurposing. So there are a lot of the drugs out there who are approved, or maybe even vitamins that could be helpful to help our kids with their symptoms. But in order to be more widely known, we need to do structured studies on them and figure out, are there toxicity, are there off effects that we haven't thought of. So all those gonna take money, it's gonna take talent, and it's gonna take time. Mary, anything to add?
Um, I know we're at time, so I appreciate your all, patience with us. I, I'm excited to hear what questions you have, what suggestions you all have. Excited to hear your input when we're finished. But this, it is a long process, and Jen has done a really nice job of soliciting folks to donate their time and energy towards this. And so, she's been a relentless advocate for Coffin-Lowry and other diseases. And I just want you to know that she's fighting for her daughter. She's fighting for all of your kids as well. And she is relentless. She's going to keep working on this. And so, I'm proud to be working with Jen on this. And I hope that y'all know that how much of her time and life she's pouring into this cause.
Yeah, thanks, Mary. I'm happy to stay on. I know we're at time, but I've cleared my schedule, so I can stay on. But welcome to drop if any of you need to. I just have a couple more slides. Um, so this is what my kids have been doing, because we know it's gonna take money and talent and time. So every fundraiser counts. I want folks, my kids and me are making jewelry. We're going out collecting $1 here, $2 there. My daughter, who has CLS, is cutting stickers and giving them out. Like, we need every one of us to work together toward a cure, because it is possible, because it has to be possible, because there are better options for my kids, for my daughter, and for your kids. And I've seen the miracle happen. Like, Mary and I work in pharma for a reason, because we know drugs do get to the finish line if we do this right. And because of it, my family is willing to commit at least $500,000 into this, just my family. So we're gonna keep doing this until it's done. Because, like I say, my daughter needs it, and probably my grandkids gonna need it. So hopefully you will all come with me on this journey. Um, so this is my concluding slide. There are, I can't do this alone. Mary can't do this alone. It does take a village, because every one of our kids is a snowflake. We need the data. We need the blood. We need your time. Anything you can contribute. We need folks to educate your doctors, right? So this is my email. It's my consortium email. If you want to directly get to me, it's jin@curendd.org. But like I say, we're absolutely committed in doing this. This is more than any job, harder than any job I ever done, but we can find a cure within my lifetime. Teresa and I go back and forth on this, but I'm like, nope, I'm confident.
All right. And we're right there with you, Jen. Hey, listen, thank you so much. And please thank your family. That is a extremely generous donation in order to help continue this research. And as a parent of a child with CLS, I greatly appreciate it. And I do have confidence that we'll get somewhere with this. So please let them know that we're all very grateful to them.
Yeah, thank you. And hopefully, we Mary and I is working nonstop. And there are more people in my team working on CLS directly. And we will do regular updates with this community. So we're, CLS Foundation is part of Kira and Didi. Teresa and I have regular calls together. So we're gonna keep pushing forward. But it's only doable if more, more of us come together.
Hey, Jen, this is Margaret again. Just a quick
Yes.
question on the funding piece. I just saw something, I'm just a layperson reading this, about California having a new initiative for rare diseases that they were
Yes.
throwing significant amounts. Is that a possible funding source?
Yes, yes, thanks for that. We're absolutely eyeing on all funding sources. So fortunately, we do have team of few PhDs, and we're working with Northwestern. So we're applying for funding in FDA, in CERM in California. We're applying for funding in NCATS, which is the entity that likes drug discovery. We're hitting several venues to get external funding to help with this.
Yeah, send us whatever you saw, and we'll make sure that we're aware of it, that it's on our list. We meet weekly with a broader team and discuss all the grants that we're working to apply for, and which grants could we potentially apply for and how. So we would love to have a look at it and make sure that it's on our radar, and if we're eligible to apply, we definitely will.
Okay.
So that goes for everybody. And if anybody has, grant writing experience, or, anybody can help with that, please do.
Okay, sure.
Yeah. Hey, Jen, I sent you something, but we haven't had a chance to talk about it yet. So I'll look and resend that email. And Mary, I'll put you on there.
Thanks.
Yeah, and if any of you know folks who's into science, who's, learning biology, like let me know, because I could always use another intern, another analyst. There's going to be a lot of data that we have to come through. So have them join our research team. Yeah, so anything, we will always try to answer your email.
Yep. And the information that Jen recently gathered, I know a few of you participated in her survey. We're planning to publish that data as well. So if you haven't already taken an interview with Jen about your experience, please do, because that also contributes to data that's publishable that we can then use to drive the field forward. Margaret, were you about to say something?
I was just going to mention, and you have probably heard about this one because I think it's been out there for a while, but Mark Zuckerberg's wife has got the initiative for rare diseases. And Teresa and I have talked about this one. You probably know this as well, but I think they give fairly large grants, but it's sort of on a 3 or 4 year cycle. And when I first read about it, it was sort of at the very end of one. They had given all the grants out for a period of time, but it seemed like that might be a natural fit as well if they open that back up again.
Send it all.
Yeah,
Okay.
yeah. Margaret, where are you based?
I'm in Atlanta.
Okay, cool. Yeah, thank you for that. I do have friends who work for CZI. Chin Zuckerberg Initiative. They unfortunately just lay off a bunch of people. So they're going through strategic arrangements right now. So we'll see. But yeah, leads like those are super helpful as well. So thanks for keeping an eye out, and for others, do send us.
Martha, did you have a comment? Yeah. Sorry, I thought I saw you talking. You might have just been talking.
Oh, did you say me?
Sorry. I did have a question, though. You talked about the biorepository. Is that something that we could do as individuals right now? Find a place that takes a sample?
Hey, Marty.
Yeah.
So I'm already working an arrangement. I've got a physician here in town. He's an internist who's agreed to do these at no cost.
Oh.
And so we've got to get our sample kits together to get this, but it's like I can't put the cart before the horse. I've got to figure out and determine which repository we're going to go with. And but yes, you are on that list for sure for North Alabama. And then I'm going to get some of the new families that are in this region. And there's some in Tennessee too that I think could come here.
So if we can help you with that at all, Teresa, just let us know.
Oh, absolutely. Yeah. I will need some help. So that'd be great. Thank you.
Okay.
I would say my biggest ask at this time is if you haven't booked a time with me, please email me. I want to listen to your stories because we are publishing that data as de-identified research to let physicians know, what are the in and out of CLS. My goal eventually is let them know um, how to better treat our kids because I've I've we just came back last week from ACMG medical geneticist. I encountered somebody who's like the only time I diagnosed somebody with a rare genetic condition without a genetic report was in CLS. It is that memorable. Like our kids have very specific facial traits, right? And musculoskeletal issues. I need that person to be cloned like in every state, right? Uh, please book a time with me. We'd love to hear your stories. And any data you can contribute will be great as a starting point.
Hey, Jen, how many families have you met with?
I've met with 6.
Okay.
Yeah. So thanks, Martha. Thanks, Annie.
Not to name names. Any other questions from the group or things that y'all think we should know?
Well, anything like you think that's important for your family that Mary and I should look into that I did not mention?
I think Judy mentioned that sides is like the worst and the most difficult to manage phenotype or characteristic. And I would second that if we could get some kind of handle on that, that's the most pharmaceuticals, that we take is related to that. And it is just debilitating.
And Julie, for your
Martha,
son,
we can't hear you. Martha,
You're
you're muted, dear.
Okay, thank you. We give Teresa full credit for Susie ever being diagnosed because we happened to be at the grocery store one day and she saw Susie fall and she said, Susie falls just like Matthew. And she told us what Matthew had. You remember that, Teresa?
I do.
For that, I was able to. Before then, I was going to taking her to doctors, and they say maybe it's Williams, maybe it's Noonan syndrome. Nobody knew what it was. So, with that, it helped us a whole lot. Thank you again, Teresa.
Oh, you're welcome. Hey, anyone else on the call have any questions?
Hello.
if not, I just want to say I will be back. We work more like this is more than full-time job. We will have updates regularly for you. And in the meantime, if there's any questions or comments or suggestions, please do email me.
Yeah, speaking of, I should also I mentioned that we did submit a grant application. We did get a tiny bit of feedback. It's a National Institutes of Health grant that we applied for. It. We feel like it's something of a long shot, but we did get feedback from them asking for more data, and so that data was due tomorrow, and we just submitted it today. So we're working hard on that, and we have another abstract we're trying to submit Monday that Jen just finished the first draft of. Thank you, Jen. So we are working all the time on this. And then I just saw a comment here, DePaul. Yes, great. Yeah, we're recording. But yeah, we're working all the time on this, and it's a small community, and sometimes I think Jen and I and the others on our team, it is a small team, but being able to share this information with you all and share our updates is meaningful to us. So thank you for listening, and please do send us whatever you can want to send us, and we are excited to continue to share updates as we have them. So thanks for giving us this opportunity to do such.
Well, I can't thank you both enough for all your hard work. Um, a simple thank you is so inadequate. But absolutely, if there's anything that any of us can do to assist, whether it be, collect more data, get out to more families, um, do more fundraising for um, our research. I feel like there is more that our community could do. I'd love for both of you to attend one of our family gatherings so you could see, firsthand, meet our families and some of the challenges that we have, because everybody's story is unique, because CLS is so different. But really, I want to thank you both for coming on tonight. I know we didn't have as large a crowd as we had wanted, but we did record this event. So I will be posting this to our Facebook group as well as putting it in on our website. So it will get out to many more families.
Super.
Yeah, thank you. Thank you, Teresa, for everything you've done for us. I mean, I'm here, because of my daughter, just like we all trying just to take care of our family. So we're not going away. And email me if there's any questions or comments, please.
Yeah, and just so you guys know, I have a monthly call with Jen, just a tag up. So we keep in regular contact, and typically we talk more than that. So
Yeah. Awesome.
all right.
Thank you
so much.
very much.
Great to
Thank
meet
you
you.
guys. This was great.
Thank you. Thanks,
Thank you,
Margaret.
everyone.
Bye-bye.
Bye.
Thank you. Thank you.